Hello and welcome to our webinar on integrated pest management techniques for commercial cannabis growers. Today we have the pleasure of hearing from Dr. Michael Brownbridge, who is the Biological Program Manager for Disease Control at BioWorks. He'll be speaking about keeping key pests and diseases at bay through the different stages of cannabis production. Michael Brownbridge has over 30 years of industry experience in production systems, biological control, and integrated pest management with an extensive research history. He previously held positions at Vineland Research and Innovation Centre in Ontario, AgResearch in New Zealand, and the University of Vermont, among others. Michael joined BioWorks in late December. In this current role, he is responsible for accelerating the adoption of current and new biological products while providing expert advice and guidance to growers. Thank you to our sponsors, TerraLink, BioWorks, and Plant Products for making today's webinar possible. To our audience, if you have questions, send them to us at any time using the questions box in the panel to your right, and we'll address as many as we can during the Q&A period after the presentation. If your connection cuts out, don't worry, because the recording will be made available online and a direct link will be sent to everyone who registered later this week. So without further ado, I'll hand the controls over to Michael.
Well, thank you, Greta. Just made me feel old when you said I've been in this industry for 30 years. Good grief. So good afternoon, everybody. It's both of you that are out there listening to this. It's really nice to be here. I do look forward to the day where we can actually do this type of presentation again in person. So as Greta said, I'm going to be talking about taking on pests and diseases in cannabis. But before getting to the meat of the presentation, so to speak, I just want to add to what Greta's already said and just say thank you to Plant Products and TerraLink for sponsoring this event, and of course to BioWorks for giving me the freedom to make this sort of presentation. So just by way of full disclosure, this is a Greenhouse Canada presentation and is primarily targeting the Canadian cannabis industry, but I know we have friends from the US as well who may well be joining us this afternoon. But by way of full disclosure, I just wanted to let you all know that I'll be specifically talking about pest and disease management tools that are approved for use in Canadian facilities, rather than just talking about a wish list of products, materials, natural enemies that could be used. And I'll also be limiting the talk to the primary production issues and solutions as they relate to pest and disease management. Recognizing that there are and there will always be more, but 40 minutes plus minus is not a lot of time to cover everything. So I have tried to limit myself to the most prevalent issues that the growers are facing. So I approached the talk and put it together not just by sort of listing all of the diseases that could happen, all of the pests that could happen, but more looking at it as perhaps a grower might look at the system when you're considering your whole production, crop production period, sort of from the start to the end. And I think that is important to do that, not only from my perspective in terms of putting this presentation together, but from the perspective of a grower, in that you have to think about, okay, think through the whole process. What issues may I be facing in this stage, this stage, this stage, or this stage? And what do I need to do proactively in terms of ordering materials? Because as this year has proven, it can be very, very challenging sometimes to get certain biocontrol agents or supplies, and access has not always been easy. So the more advanced planning you can put in place to your IPM program, the more advanced warning you can give to your suppliers around needs, the better. So I wanted to approach this because I think sometimes there are unique challenges in certain stages or unique approaches to a specific crop development stage. So I approached it from that perspective. I will also be focusing on indoor production. That's in a greenhouse or a grow facility. But if anybody is tuned in today and is growing outside, I think the propagation methods certainly that I'll touch on today could be applied to production of transplants for either outdoor cannabis or hemp. So I'm also came at this from the perspective of putting together what I'll call a functional IPM program for cannabis. And yes, I'm going to limit my comments to the use of primarily to the use of biopesticides and biological control agents, rather than trying to cover all of the cultural aspects as well, which is, in my way of thinking, that's a whole other webinar or three. And I tried to put the talk together considering the best fit of the components that are largely compatible or can be readily integrated together into a crop protection scheme. Having said that, of course, I'm going to go into nutrition, but I do think it is so important that we do touch upon this and think about the importance of plant nutrition from the outset. It does have a direct impact on plant health. And by optimizing growing practices, we can do an awful lot to create a more resilient crop that is not only more productive, of higher quality, but it's inherently less prone to pests and diseases as well. If I look at this, you know, I spent most of my life in the last sort of 10, 11, 12 years working in greenhouse ornamentals and vegetables, for example. And certainly on the ornamental side, we know that many greenhouse ornamental crops are actually over-fertilized. Fertilizer, by and large, is pretty cheap, so there's no downside to it from a cost perspective. But we know that high nitrogen levels in plant tissue in particular can actually have a very dramatic effect on that plant's susceptibility to pests and diseases. And this is how plant nutrition really should be considered as part of your IPM program. And this link between high nitrogen and crop susceptibility to pests has been previously demonstrated in a variety of field crops, ranging from cereals to vegetables and fruits. And studies have documented a particularly strong correlation between high levels of applied nitrogen fertilizer, that's synthetic nitrogen fertilizer, and growth of pest populations. And I'm sure you'll recognize some of the pests listed in the table as common not only in a wide variety of greenhouse crops, but also in cannabis. And the fact is that populations of pests such as two-spot spider mite, green peach aphid, and western flower thrips will expand rapidly when high levels of synthetic nitrogen are applied. I mean, that raises an intriguing question: is that can we alter fertilizer inputs? Can we reduce them without affecting crop quality or production time? And in so doing, can we also reduce fertilizer rates as a means of slowing pest population growth? And if we can do that, then that just supports the greater goals of an IPM program in terms of preventing pest populations developing so rapidly. And we know that in a crop, well, all crops oftentimes, but particularly in cannabis, manipulation of essential nutrients like nitrogen, phosphorus, potassium is required throughout the life of any plant. And in cannabis, there's oftentimes a push at flowering to produce larger blooms, but that can also compromise plant tissue formation, so that those flowers are actually, they may be bigger, but they're also more susceptible potentially to powdery mildew and botrytis. Now, with recent changes to the Health Canada regulations around the application of nutrients by foliar spray, that really brings more options to the table, that we can actually now potentially reduce salt-based nutrient inputs, so nutrients applied through your irrigation scheme, to using perhaps some foliar applied nutrients without compromising yield. So we can reduce those salt inputs, salt-based nutrient inputs, and use the foliar inputs. And we know that research in other crops has previously demonstrated that application of foliar nutrients at times of peak nutrient demand, and when are times of peak nutrient demand? It's like when plants are rapidly growing, when they're going into bloom and flower development, as well as prior to stress events such as drought, temperature extremes, or major changes in light and humidity. And if we can provide these key nutrients at those times, then we can enhance plant stress tolerance. And use of some sprayable nutrient supplements, if you will, can actually also enhance uptake of elements like calcium, which, you know, in poinsettia crops, applying calcium chloride by spray, as a foliar spray, to improve uptake at certain parts of the plant where it's really hard to get calcium into those tissues, and particularly into cell walls. And we know calcium is an important component of plant cell walls. The bottom line is that stronger cells, especially in flowers, can reduce susceptibility to diseases like botrytis. And this, as I said, has been demonstrated in petunia, whose flowers are very susceptible to botrytis. And that susceptibility has been linked to poor movement of calcium into the flowers. And those deficiencies result in weaker cell walls. That translates to increased susceptibility to botrytis. It's not yet been proven in cannabis. There is work underway to do this, but we believe it's likely that enhanced levels of calcium in blooms will be one of those things that can help us to control botrytis. Again, it gets back to my comment that, you know, plant nutrition is an absolutely fundamental part of an IPM program, and getting that right is essential. So moving on, you're going to get absolutely sick and tired of me saying terms like sanitation and prevention today, and the importance of both in pest and disease management. And this is really particularly the case when utilizing biological inputs as cornerstones of IPM systems. As we know, you know, at its core, biological control is a preventative and not a curative strategy. And biocontrol agents, and by that I mean predators, parasitoids, and biopesticides, really should be applied to best effect when those applications are made proactively at this point, rather than later during that sort of exponential growth phase of a pest population or even incidence of disease. And it's particularly important in cannabis because there's no get out of jail free cards when you're growing cannabis. There's no rescue products, at least legal ones, that can save a crop when pests or diseases get the upper hand. So use of good sanitation and prevention practices, that's your foundation for all pest and disease management in cannabis. And it's not me that says that. If you're familiar with Alison Justice, she's really one of the pioneers in hemp, certainly, but also in cannabis production, and really one of the very, if you just Google the name Alison Justice, you'll find out she's done a lot of great work in this area. So what's true in life is really also true in cannabis. Look after your mother. Sanitation starts from the very beginning, and if you maintain your own stock plants, it's a great position to be in. It can be made better or best, if you will, if this area is dedicated to keeping these mother plants healthy in a stress-free and clean growing environment, which ideally has systems and mechanisms in place to prevent ingress of bacterial and fungal spores and insects. And then limiting access to this dedicated room further reduces the risk of transfer of pests and diseases into that room. We know that people are a primary source of movement of pests and diseases inside a greenhouse or a grow facility. So restricting plant care to a few individuals can go an awful long way to reducing the risk of transfer of those diseases and pests into that room. I would stress that plants should be taken good care of. This is your investment. Test them regularly to ensure they're free from viruses and other diseases, and stay on top of bioprograms. And because you're not actually growing these plants to produce a flower, then you can use sprays on these plants pretty regularly, approved sprays that is, so you can keep them free from pests, diseases in a different manner to methods you would perhaps use in your actual cannabis production facility. And as long as you stay on top of those, then I'm suggesting that you know, the clones that are taken from those plants should also be of good health, because diseases and pests do transfer on clones. This we know. And there's various steps you can take to improve the sort of nutritional health of those plants, and that nutrition is transferred to the clones so that you can get better rooting, faster root development, and stronger clones from the get-go. So here we are. There's that sanitation word. I'm sure when I said sanitation, many will perhaps think about this solely being applied to production space, for example, and certainly growing spaces and rooms should be thoroughly cleaned, and that includes removing any and all debris and sanitized after a crop is harvested and prior to a new crop being brought into that space, thereby reducing the risk of pests and diseases carrying over from one crop cycle to the next. But the good sanitation practices really should be applied in every stage of production. And use of materials, hydrogen peroxide-based materials particularly, these are, I think, really useful and versatile materials to use particularly in the cannabis sanitation strategy, because they can be applied both to hard surfaces for sterilization of hard surfaces, but also to plants. And what varies is the dilution rate that you use from those products. So the goal of every grower should be to start a crop clean, as this does reduce potential problems later in production. And it starts with the mother plants, as we've already discussed, and that extends into the propagation of clones, how they're taken, how they're managed. Now, peroxides can be used to sanitize tools and surfaces, as well as any flats or trays used for those young plants. Used to sanitize irrigation lines, which can harbor diseases in biofilm layers, which inevitably build up inside them. And in addition to that, it can actually be drenched onto the rooting medium or rooting cubes, depending on what you use, to sterilize the medium or the cubes. Apply it the day before sticking, and that essentially gives you a nice sterile environment in which to stick your clone. Even goes so far as to say sort of dipping clones in a dilute peroxide solution is a great way to eliminate surface contaminants from that clone. It's just one more step to sort of sanitize plants from the very beginning. Now, what happens if you're bringing in clones? And I know many facilities have a sort of a hybrid, and that they produce some clones in-house, but they oftentimes bring in clones. And I've heard quite a few times now of greenhouses claiming to have, for example, we never had root aphid issues until we brought in clones from. So sanitation is really doubly important here. And at some point, especially with the recent incidents of virus-infected clones arriving from certain propagators, I'm sure there'll be calls, or just it's a matter of fact that the industry has to reach this point, that there has to be some sort of phytosanitary certification accompanying the material. That's good. You shouldn't always rely on that. So until that time, and let's say even after such time, sanitation is always going to be your friend. Peroxide dips again, or dips in biopesticides such as Botanigard, are ways to reduce or eliminate microbial contaminants or insect pests. Are you able to quarantine the clones so that they're produced in a separate sort of room away from your primary production area, such that there is if there is an outbreak or an infestation there, it doesn't transfer from the clones to the crop, and ditto, you don't want to be transferring from the crop to those clones. Again, limiting the access of personnel, having a specific sort of order of entry plan in place can go an awful long way to reducing transfer of pests or diseases in and out. And I think one important thing to do as a matter of course is sending out tissue samples for analysis, so you'll get results back pretty quickly that you can then have confidence that your young plants are free from certain diseases, and definitely from viruses. Of course, there's a cost to it, but if you weigh the relatively minor cost incurred there compared to the major costs of control or crop loss, then it's a small investment in the future crop. So after taking care of mother, you have to nurture your babies. So prevention, as I said, starts right here. Nipping pests and disease problems in the bud, no pun intended. And you also have the opportunity here to promote healthy root development with how you have taken those cuttings, how you feed those cuttings. And as we know, healthy roots typically equates to a healthy plant. So managing root diseases is part of doing business in any greenhouse crop, really, and that includes cannabis. And the usual suspects that infect other crops can also infect and compromise, or in severe cases, kill cannabis. Pythium and Fusarium species, these are known pathogens on a range of other economically important greenhouse crops, and they're quite happy to use cannabis as just another host. And unfortunately, they are readily transferred in irrigation water, soil, and on people. And yes, they can come in via infected clones. So be aware of the diseases. So what can we do? Comes down to good sanitation practices. I'm sounding a little like a broken record here, but disease-free clones ideally. Testing your water supply to ensure it's free from any of those diseases. Putting sanitation or sanitization processes in place to sterilize your water prior to it going into the crop is important. And testing the water before and after that water sterilization process, that tells you what's there and how well your processes are working, such that you're actually feeding in clean water to your plant. And then there's various preventative tools, biological tools that you can use to prevent the diseases. And the fungi and products like RootShield, RootShield PLUS, and Pre-Stop, for example, will colonize and grow on developing roots, and in so doing, will protect them from many soil-borne pathogens. And these fungi, these trichoderma fungi or glioculadium fungi, actually bring several different modes of action to the system to protect roots. Trichoderma and glioculadium, for example, have a combination of various mechanisms they bring. Antagonism, where they produce materials that inhibit growth or even kill other microorganisms. Competition, where they outcompete pathogens for space and nutrients on the roots. Some can even actually predate or parasitize soil pathogens, and then induce resistance, which means that the presence of these fungi on the roots actually turns on some of the plant's own defense mechanisms. So a combination of all of these mechanisms are typically brought by these biopesticides. Often other plant benefits to using these biocontrols. There's a lot of research documenting the benefits of say, trichoderma inoculants, such as stimulation of root growth, enhancing access to soil nutrients so that they're more accessible to the plant, and that translates into promoting plant growth and health, and certainly improved development through the life of that plant. And certainly in trials done on RootShield Plus, for example, in cannabis, application just at sticking resulted in improved root development in clones, and that ultimately translated to a 4% increase in yield of flowers at harvest. So you can see how something right at the beginning of a crop can actually translate all the way through to benefits at the end of the crop as well. So plants with a healthy start in life do perform better. So moving on to pests, there are many, but I'd say those of primary concern in propagation are mites, aphids, thrips, and fungus gnats. So there's three mites I would say are probably the most concerned: the hemp russet mite, broad mite, and two-spotted spider mite. And yes, alas, all can come in on clones. Commonalities across these include a high capacity for population increase. And owing to their small size, they're oftentimes hard to detect, and so that the first sign of an infestation is often damage symptoms. So by that time, it's probably too late to effect control, certainly with the tools that we have available to us. So as we only have the tools to prevent pests from increasing. So you really have to act early through the use of biopesticides, I would say, to produce that sort of quicker knockdown, reduction in pest or numbers or in diseases, and then supported by the early implementation of biocontrol strategies. So let's have a look at some of the options we have. Mineral oils can provide control of several problem pests, including mites, and they can control all stages of mites, including eggs, thrips, aphids, and whiteflies. And there's 2 products really approved for use in cannabis, Sufoil X and Pure Spray FX. They're both based on horticultural oil, but the formulations are quite different, and those impart different characteristics and different performance benefits to each product. And as a note, a side benefit of something like Sufoil X particularly is its ability to suppress powdery mildew. So applying this to control pests and pests also sort of complements use of biofungicides. So application rates generally are in that 1 to 2% range, similar for both products, with reapplication after 7 to 14 days. And as they're both contact insecticides, thorough spray coverage is absolutely essential to efficacy, but do not spray to the point of runoff. Also, have to be a little careful when you're applying to young rooted cuttings. It's probably best to go in with a 1% rate until you're pretty confident that there's going to be no harm to your crop, and ideally wait until the plants have developed roots before spraying. So sometime around 70 days after sticking. Test for phytotoxicity if you're a little unsure, you've never done this. And just note that the use of spray oils will leave a sheen on the leaves, but that does not interfere with any transpiration. Just some precautions around use of any oils. You have to be sure to use them when the relative humidity is less than 90%, temperatures below 33 degrees, and you really want these materials to dry in no more than 2 hours, because they're oil-based and the cuticle of plant leaves is waxy. If the oils are allowed to sit on that cuticle for too long, they can start to degrade the waxes on that leaf surface. So biological control agents, or BCAs, available include Andersonii for hemp russet mite. It's okay. It's not brilliant, I would say. Oils are probably really something you really need to use to manage hemp russet mite, at least initially. For broad mite, Cucumeris does a really great job against broad mite and 2-spot spider mite. Phallacis at this stage in production is an excellent way to go. I mentioned around for pests like hemp russet mite, use of oils. It does raise questions around compatibility. We know that oils will also kill predators on contact. But if you're using predators, particularly if you're releasing them via sachets into the crop, once the spray residues are dry from these spray oils, they have no toxic effect, and they won't harm the mites in the sachets. Right, try that one. Say that one quickly 3 times. It's impossible. Rice root aphid is another pest that can get quickly out of hand unless you get on top of it early. Drenching rooting blocks with Botanigard 22 WP is a great tactic. This was recently approved again through Health Canada PMRA as an approved application technique. It's important to make sure the cubes are thoroughly wetted, so you're getting the material throughout the cube, throughout that where the roots are growing and into contact with the aphids. And it's important to repeat as well. It's not a one and done approach. And later in the production, really, you need to accompany root drenches with foliar sprays to target adult root aphids, which will emerge from the substrate. Western flower thrips, yeah, this is a universal pest. Having spent a good part of my career working on Western flower thrips, I had a certain fondness for it, but I also have a certain fondness for all the agents we use against it. And there are quite a few that can be used in cannabis production. Do you need to use all of them? Probably not. It really depends on what works for you, and it depends on the level of infestation. Some of these agents will also, of course, remember this, provide simultaneous control of other pests. Cucumeris, for example, will concurrently control broad mites. Stratiolaelaps and Deloisia will also feed on fungus gnat larvae. You also need to remember that for a pest like western flower thrips particularly, you need different strategies to control those different stages in the thrips life cycle. So there's the foliar feeding stages, that's where you use Bavaria products and Cucumeris, and then pupating stages which occur in the substrate, and that's why you need soil-dwelling predators like Stratiolaelaps and Deloisia. And of course, these things will move with the plants when they're potted up into the main production area. Fungus gnats, maybe they're more of a nuisance problem, but we do know that they can passively vector soil-borne diseases such as Fusarium, the adults, and at high numbers of larvae in the growing substrate, they will feed on plant tissues, and they can cause significant feeding damage to freshly stocked clones. The best option is to keep them out, but if you have to, there are various options. Starting to see a lot of repeats now, as I said. Stratiolaelaps and Deloisia will work well against fungus gnat larvae as well. They predate on those just as they do western flower thrips, so they'll manage these soil-dwelling stages, these fungus gnat larvae. And if you have a really bad infestation, you could also consider using nematodes. Steinonema feltii is the one to use. And again, the overlap here that Steinonema feltii will also control pupating thrips. And one thing I think we sometimes forget about is good old yellow sticky cards. You should have them up to monitor for pests, or for flying stages, for pests that have flying stages, but they're also really, really useful. They can make a significant contribution to the overall removal of adult pests like fungus gnats and thrips. So they're a good thing to have up, especially in the production space, which is typically a smaller space, but having them up there, they can, certainly trap out a lot of those fungus gnat larvae. Fungus gnat adults. So moving out of propagation with plants now being potted on and moving into the vegetative phase of their growth. Pests and diseases, which are a threat during propagation, remain so at this stage of the plant's development. And steps taken in propagation will carry through this stage. And you'll hopefully have everything under control, but you can't afford to relax. And that really means continuing a lot of those practices that started in that propagation stage now into the production space. And for example, while cultural practices like pruning and air circulation now come into play to keep foliar diseases, which weren't necessarily a problem in that propagation phase, but foliar diseases like powdery mildew, use of good cultural practices can help keep that in check, but it still frequently shows up. Again, as there are no curative controls, it's vital to stay on top of things. And again, here we are, use of the preventative tools that we have. Crop scouting is essential to spot early signs of infection. And the example I have here is if you look at those cannabis leaves carefully, you'll see some little marks on there. I just use those arrows to highlight where they are. That's early stages of powdery mildew. So you've really got to be looking at your crops to detect that early before it reaches this stage where it's way harder to get control of that. And similarly, it's essential to scout your crops so that you can take timely actions to keep pests in check. Fortunately, we do have some excellent products approved for use that allow to preventative control programs, as I said, to continue essentially in a seamless fashion through production. And that includes, again, use of fungal biopesticides applied to that growing medium. Things like RootShield, RootShield PLUS, pre-stop. Again, it's important to drench the pot or the growing container of any sort with sufficient suspension to treat the entire pot. You don't just want to drop in a few mils, which really will only treat one small portion of that growing substrate. You want to be able to treat it also all the roots are protected. And you can reapply if you're using the wettable powder formulations. You can reapply these through the irrigation system after 3 to 6 weeks, depending on the product. Another option, if potting up or growing in a sort of a peat-based or medium, is actually incorporating granular formulations into that substrate as well. And that's where RootShield granules, for example, the RootShield Plus granules can be a really good and cost-effective way to go. And again, you know, healthy roots means a healthy plant, so it's important that we protect them. For foliar diseases, at this stage, you know, this vegetative growth phase through to early flowering, I think powdery mildew is one of the biggest challenges faced in pretty well any facility. And again, there are some really good products out there. There's potassium bicarbonate products are available for use in preventative or curative manner. This is probably one of the few biopesticides that can be used in a curative manner. And the nice thing is potassium bicarbonate has a physical mode of action in that it desiccates mildew spores, it destroys the cell membranes of the fungal mycelia, and it alters the pH at the leaf surface, so that becomes really not a great environment for fungi to grow in. These can be a little harsh on soft-bodied biocontrol agents, but again, they don't harm predators in the sachets, and once the spray is dry, the residues are not harmful. Regalia Max biofungicide is a different type of biofungicide. Again, it's a liquid concentrate based on an extract from the giant knotweed, and this one works by inducing a resistance response in the plants it's applied to, and it's the plant's own defense mechanisms which protect it from infection by powdery mildew. And these products, Regalia and potassium bicarbonate, like Milkstop or Sirocco, you know, they are compatible. Great to use them perhaps in a spray rotation, so that you get different modes of action to bear in the crop. Again, stepping up from that propagation phase, you see the list of pest threats in that crop has grown a little bit longer, and now includes other aphid species, what we call the small species, green peach aphid, cotton melon aphid, and the large species like the cannabis aphid, potato aphid, foxglove aphid, as well as the other suspects that we saw as a potential threat in the propagation phase. I've included whiteflies here just to be aware of it. I'm not going to really focus on it today. There are controlled parasitoids, particularly that can be used against whiteflies, but typically speaking, they're not a common pest in northern greenhouses. Biopesticide options include, again, the mineral oils for mite control, Sulfoyl-X, Pure Spray FX, where rather than spraying the whole crop now, and this is again where crop scouting is important, is coming in and applying them to hotspots so that you can preserve natural enemies which are used in the wider crop. Remember, they have a 12-hour REI associated with them. Similar precautions, but one thing perhaps that we may be seeing here is using them together with, I know sulfur is often used to prevent powdery mildew, but also to effect mite control. Well, these mineral oils, you've got to be super careful about using them if sulfur's been used as well. So there are some concerns around the phytotoxicity if they're used too closely together. So be aware of that. And again, it comes to sort of thinking that whole program through. What am I going to use to control what and when, and how compatible are the different components of this program? Again, make sure that you're applying at a time of day when that spray will dry in 1 to 2 hours, and don't fog it. And the reasons behind that is that fogging requires use of pretty much the same amount of active in a way smaller volume of water, so that you're exceeding the rate concentrations in the material applied, so you're increasing the chances of there being phytotoxic effects on those plants. The fact is that these mineral oils will also control things like aphids and thrips. So again, you get additional benefits from their use. Also, there are insecticidal soaps that are available for spray into this crop at these stages. Things like copper insecticidal soap and opal insecticidal soap at a 1 to 2% rate. Western flower thrips, they can be there. Look for them. You'll oftentimes see feeding damage scars on the leaves as perhaps the first indication. But these are times when, yes, again, using the Beauveria bassiana sprays, Botanigard or Bioceres foliar sprays can work really, really well to keep those thrips populations in check. But here's a time when really I think it's a good time to start using entomopathogenic nematodes as a sprench or drench applied to the growing medium to control pupating thrips. For the biocontrol agents, Cucumeris, Stratiolaelaps, Deloitia, these are ones which you know, we've used all of these previously, perhaps. So and these will carry at least the predatory mites, the Stratiolaelaps and Deloitia. They will carry through from that propagation phase into the main production. So you're adding in the main sort of vegetative production phase, you're helping those populations become established. And you shouldn't need to apply these repeatedly. They should become established in that crop and provide long-term control through the lifetime of that crop. The newbie, I think, primarily to this equation is aureus, ideally released into the hotspots to become established. Just one note here that it's probably not the best biocontrol agent to use in indoor grow facilities. They like natural light as opposed to artificial lights. And they're prone to diapause in the shorter daylight months, so November through to February, for example. And as I said previously, many of these biocontrol agents will also keep fungus gnats in check. There's nothing specific to fungus gnats that in terms of natural enemies that is unique to their management. Just again, cultural practices, good watering strategies, keep those upper layers of the soil dry-ish so that it's not conducive to fungus gnat growth. I just want to perhaps harp on about this, that it's so important to get spraying right, particularly with biopesticides, because they are contact. They work by contact, either with the pest or the disease. So it's absolutely essential that you get good coverage of contact. And there's many makes and models of sprayers available, but they essentially fall into two types: hydraulic sprayers and low volume sprayers. And they use different methods to produce and deliver spray droplets, and those spray droplets have different sizes. So understand your spray equipment. Really, absolutely invest in quality, reliable spray equipment. Not only is that equipment built to apply pesticides, but you'll get more uniform coverage. You will apply what you think you are applying within a given area. And we know for a fact that sort of incorrect timing and poor coverage are really these primary causes for biopesticide failure. And use of improper equipment, I should say, or not sticking to best spray practices is one of the main reasons for phytotoxicity on that crop. Of the biocontrol agents, again, for hemp russet mite, Andersoni can be used this time. Instead of using the sachets on a stick and one per tray, we're actually hanging the sachets in the plant. Similarly, for Cucumeris, for broad mite control, we're hanging those sachets in the plant. We're getting also a thrips control using those Cucumeris. And then for two-spot spider mite, the additional natural enemy you may want to use, you may have to use here is Phytocoelis pessimilis, and this is a phenomenal predator, very compatible with Phalasis if that's been used in prior stages of production. But this one's a really good tool to have. You start seeing hot spots in your crop, release bacillus into these hotspots, and it can be used to really clean up a two-spot spider mite infestation in those hotspots. Again, you may have to release it more than once, but again, reiterate, scouting to detect these problems early so that you can respond early. For aphids, now we have quite a few, as I said earlier, more aphid species to think about and be able to recognize. And it's important to be able to recognize those aphids. So do a crash course in the quick and dirty course in aphid taxonomy, because different aphid parasitoids will parasitize different aphid species, or they'll parasitize some better than others. So if we divide the aphid pests into small aphids, green peach, cotton bean aphids, and large aphids, foxglove aphid, potato aphid, cannabis aphid, for example, then there's different aphidia species which are needed for these different aphid pests. Predators, a little bit more agnostic in what they'll eat, and these are more generalist predators, Aphidoletes, for example, and Chrysoperla or lacewing larvae. They can really do an excellent job of cleaning up infestations. Just a note again about use of Aphidoletes, they really don't establish or do super well under artificial lights. They prefer to function under natural light. So if you're having problems establishing in artificial lighting conditions, then you may need to rethink your aphid control program. Rice root aphid requires a different approach. They're on the roots. So we're using, again, recommending use of drenches to control rice root aphids here. And they can be used in different ways. You can vary the rates used depending on the level of infestation. You can vary the frequency at which these applications are made depending on the level of infestation. The one thing is, it's not, again, you can't just make a simple one application and walk away. What is important is that growing medium is saturated so that the Bavaria in Botanigarda Bioceres reaches the entire root ball, reaches all of the aphids in that root ball, because contact, again, is essential. And at this point, when we're in this production phase, this vegetative growth phase, it's important to pair the drenches with foliar sprays so that we're also targeting the winged adults. And that's essentially a 1-2 punch, targeting different life stages in the root aphid life cycle. And that improves overall efficacy of the program. Now, when we're moving sort of from flowering and into harvest now, this is where the challenges really start, because we start to see that we have reduced pest and disease management options available to us. And this really comes for two reasons. One is that we have, quite a few microbial sprays that we can use, but you don't want to be spraying microbes when all of the crops have to go through a microbial analysis after harvest. So pulling back about 3 weeks prior to harvest on the use of microbes is prudent. Potassium bicarbonate, stopping the sprays of potassium bicarbonate to avoid risks of damaging the trichomes is important. So again, we're losing a valuable tool in our arsenal here if we have to stop the potassium bicarbonate sprays. Sulfoyl X, maybe slightly softer, but again, don't use that. I would not recommend using that right up to harvest. Some growers have used it with good success, but probably I would back off 1 to 2 weeks prior to harvest. But if you're on top of everything, as you should be, sanitation prevention, pests and diseases should have been controlled prior to that final 3 weeks. But if we have to, well, what do we have available to us to prevent diseases like powdery mildew and botrytis at this stage? Well, this is where Regalia Max, a biofungicide, can be used. RootShield HC as a foliar spray. This is again based on trichoderma, spraying the foliage every 1 to 2 weeks. Similarly for the Gliocladium product, pre-stop, spraying every 3 to 4 weeks perhaps. But again, these are both contain living microbes. And as I said, potassium bicarbonates like Millstop and Sirocco through those early flowering stages only. None of those products will give you guaranteed botrytis control, but they all will help with suppression of botrytis. I think one of the few tools you can use up to sort of the day of harvest practically is hydrogen peroxide. So products like xylitol here, as I said, these are very, very versatile products. Can be used throughout this production of a crop like cannabis from the very beginning in propagation and during production as part of that preventative phytosanitation strategy. They can even be used as foliar sprays. And then it's one of the things I think is a really unique feature of these is that can actually be used into that flowering period, and they're like 1 to 2 weeks prior to harvest. They can help reduce the sort of microbial loadings in those flowers. And then right at the end, cleaning up the space after the crop, that whole cleanup and sanitation of that whole growing facility. So take homes, limited number of options available to us, no rescue products. So you have to rely on sanitation and use of preventative strategies. Look after mother, look after the children, and apply preventatives early. And I would say early and heavy right up front. Where you want that mantra is start clean and stay clean. So with that, I hope I'm not too late in the process. Just want to say, thanks for your time, thanks for your attention, and happy to take any questions if there are any right now. Greta.
Well, thanks, Michael. I gotta say, your 30 years of experience really shows. It's a good thing.
Bless you.
Okay.
There will be chocolate in your next care package.
Oh, looking forward to that.
You got it.
So let's tackle some questions from the audience, shall we?
Mm-hmm.
First one, what late flower options do you recommend for thrips if biocontrol failed to control it in the vegetative and early flower stages?
Yeah, once you get late into that stage, I would say you really don't, if it's really super late, just ask, are thrips getting into the flowers? Is it a problem in the flowers themselves? The only options you have would be, I think, soil applied solutions at that point, where you're targeting your pupating stages of thrips. It's a tough one. There are, depending on how far you are out from harvest, let me put it another way, the closer you are to harvest, the fewer options you might have. But it really comes down to, as I see it, the only options of foliar sprays for thrips, like Sofoil X, like Botanigard or Bioceres, like soaps, and perhaps increasing the frequency of those sprays. But as I tried to convey towards the end of the talk, the closer you get to harvest, those really cease to be viable options because of the risks to the flowers, and you just don't want to be applying them so that they get into the flowers. So tough question. If they fail, there really are, as I said, there are no rescue solutions. So that's why it's super important to get on top of things as much as possible through those early stages. And if you have to invest a little more up front, it's going to pay dividends down the line.
Speaking of thrips, we have a question about whether or not cucumeris is a good option as well.
Cucumeris is a great option for foliar stages of western flower thrips. There are other predators. Swirsky is an excellent predator of thrips. It probably consumes more thrips than cucumeris does. It works better at sort of higher temperatures. So going into summer, it is something you might want to consider shifting to. Now, herein lies the rub. Cucumeris is way cheaper than Swirsky, so you may want to go the alternate route and sort of release more cucumeris into the crop. Again, this is what works for you, the individual, you, the grower. And it may be that you start with cucumeris and then switch to Swirsky to manage thrips, foliar stages of thrips. Swirsky, I think, also will control, not just the first instar stages, but it also goes into the second instar. Challenge this year, I'm sure you're all aware that there have been problems in actually getting hold of Swirsky from certain suppliers. because there's been issues in the production of Swirsky. So having a plan B is always a good thing. So if you're planning on using Swirsky, it's not available, then curcuma is really it's a viable alternative. But if you don't think it's doing the job for you, then try you consider increasing the rates. And again, that's something you should have that conversation with your crop consultant or your supplier. They should have that knowledge available to you.
Okay, and a follow-up. So we've just been calling them thrips.
Yep.
One reader was wondering if the tools you mentioned for Western flower thrips can be applied to onion thrips.
Aha, I bet I knew who sent in that question. That is a good question. I personally think that a lot of the tools will function against onion thrips. What we don't know yet is how well they function, because and a shout out to a young lady by the name of Ashley Summerfield, who's doing her master's thesis on this, is looking at ways of managing onion thrips compared to Western flower thrips. To do the same tools we use for Western flower thrips work as well against onion thrips, and she's doing a master's at the University of Guelph. So Ashley, I hope everything is going really, really well for you. So we will have answers to that question, but we don't know. Onion thrips and Western flower thrips differ subtly in terms of their biology. Certainly in floriculture crops, in cannabis, I don't know how different their biology is in cannabis. Is it so different that the approaches we're taking for Western flower thrips just sort of miss the mark as far as onion thrips are concerned? I would say we don't have enough knowledge, but I would sort of say until that time, I would continue to look at use of generalist predators like Swirsky. I'm going back to Swirsky, but cucumbers as well, but certainly Swirsky, and having those present in the crop. I think using nematode drenches certainly should work as well, and fungal sprays. Certainly we have some limited experience with thrips bassii or onion thrips in terms of their susceptibility to Bavaria bassiana. We know that they're reasonably susceptible. So long answer to a short question. Do what you're doing. Again, extra vigilance. Be able to identify and differentiate between Western flower thrips and onion thrips. And there's a great taxonomy, grower taxonomy tool that's available on the Floriculture website for free. So a shout out to Sarah Jendrisic there and Ashley Summerfield for putting that together. Really good job. But it uses very simple taxonomic features on these and other thrip species as well to differentiate between the two. So at least when you know what you've got, you can perhaps adjust your control strategy accordingly, depending on the prevalent species that are present.
All right, well, we actually have a ton of questions waiting for you, but I think we'll just take one last one, and we'll answer the other ones by email and post them online.
Yep.
How do you ensure that there is no cross-contamination of pests and pest control agents between rows when growing in large grow rooms?
You can't. I think that's the nature of the crop. Once those crops are touching, once people are touching one row and then touching another, things can transfer from one side to the other quite happily. Mm-hmm. I understand why crops are grown at such a high density, but it's also a downside sometimes of the high density. And I touched on it very briefly. If I've got a couple of minutes just to sort of wrap up my thought on this one, is that we always think about pests coming in from the outside. They sort of come in through vents and this and that and the other. Well, pests and diseases, for that matter, can come in on people too, just to force that point home. And I guess a lot of folks may not only be working in a facility that grows cannabis, but also growing cannabis at home. And sometimes, the pests and diseases you have at home can travel to work with you. That's bring your pests and diseases to work day. So these are things to be aware of. And you know, when you're putting together a sanitation strategy, how does that affect people as well? Rather than just thinking, you know, the point I tried to make earlier was sanitation is not just about structures, media and water, it's also about people. So bear these sort of things in mind when you put together that strategy.
Well, we'll definitely follow up on all of these questions. But that's all the time we have for today. So thank you to Michael for sharing his expertise with us. And thank you to TerraLink, BioWorks, and Plant Products for making this webinar possible today. To our audience, thank you for joining us. If we didn't get to your question, and we likely didn't, we'll post the answers online on our website in a couple of days. And a recording of this presentation will be made available online later this week. A direct link will be sent to everybody who registered. So thank you for joining us. Thank you, and until next time.
Bye-bye.
Okay, welcome to a special extended Q&A for the webinar on tackling pests and diseases in cannabis that was presented by Dr. Michael Brownbridge earlier today. We had a lot of questions that we weren't able to get to, so we're going to take the next hour to tackle as many of them as we can. So first question, which nutrients would you use on mother plants 48 hours prior to taking cuttings?
Yeah, you see, I was very subtle in my placement of product in that slide. So if you look at the slide carefully, it's there. Actually, full disclosure, I work for BioWorks, and this is a BioWorks product, but we have two products. One called On Guard, and the other called On Guard Calcium. And On Guard is it's a protein hydrolysate, in that it's enzymatically dissociated soybean protein broken down into amino acids, which can be absorbed very readily by the plant. That's why it can be applied as a foliar spray, taken up by the plant very, very happily. We have another product called On Guard Calcium, which is the basic On Guard slightly lower well, let me go back. On Guard is a 500. It actually contains nitrogen and a few other elements as well, but long story, it's all to do with how fertilizers are described and approved. Not going there. But then we have one called On Guard Calcium, which is basically On Guard supplemented with calcium chloride, because the On Guard has been shown to enhance uptake of calcium chloride by certainly in ornamentals, and that's, you know, where the example I think I gave for petunia, but it's also been done in poinsettia, also been done in lettuces, because these crops can either be growing so fast, like lettuce, that it's very hard for all the nutrients to reach those leaf margins, and that's why you sometimes see the brown necrosis, the brown edges on lettuce leaves, or it's because the calcium doesn't get all the way, so the cells don't develop as strongly, they die. That's where you get the necrotic tissue. Anyway, you've got dead tissue, it's a point of entry for pathogens like botrytis. The On Guard Calcium is useful because it also, I'm going to back up another step, I'm sorry. The On Guard used alone with a calcium chloride spray, that movement of amino acids into the plant also takes along calcium chloride, calcium, into the plant cells. And that's why applying as a foliar spray means you're getting the nutrients exactly where you need them very, very quickly. Now, in the case of the mother plants, this specific question, I would contemplate spraying them about 48 hours, 72 hours, or even applying by drench. It's also very easily taken up by the roots, totally up to you. But what it does, it works as a biostimulant almost, in that it enhances accumulation of carbohydrates in the plant, in the leaves, in the leaf petioles. So when you take cuttings, essentially as our nutrition guy, and he probably would be going like this with me trying to answer this question for him, but this is what I've gleaned by osmosis, and he's super good at what he does. But essentially, you're carbo loading the plant, so that when you take the cuttings, they're also carbo loaded. Means that they have a lot of energy resources available to them. So when you stick them, that energy is there in the plant and goes directly into root formation, plant growth and development. So you typically see superior and faster root formation and growth when you apply that foliar feed. The second benefit is actually to the mother plant itself. These foliar feeds work super well in terms of enabling the plants they're applied to cope with stress better. I've considered spraying myself with it. It works that well. I'm joking about that one. But when you apply it to the mother plants, they're coming into a stressful time. The stress being the removal of those clones. So the more nutrient competent the plant is, the better able it is to deal with that stress, and the better it will bounce back from that stress as well. So you're protecting the health of the mother and the child, to extend that analogy yet again, by applying those foliar nutrients to the mother plants. And that then applies through the whole production phase as well. But cannabis nutrition is a whole ball of wax on its own that it should be addressed at some point. It may already have been somebody talked to in one of these webinars, but essentially using these nutrient supplements, if you will, which, as I said, you can put on by drip, or you can apply them as part of a tank mix with something, or you can apply them on their own as a foliar spray. If you apply them through the plant's life, the plant is better equipped to deal with stress situations, whatever that stress may be. And that includes being better able to tolerate pests and diseases too. So it's a whole, I'm not going to repeat, I already said it at the beginning of the webinar, but it is super important. And but using foliar sprays avoids applying synthetic fertilizers, so you don't risk getting that salt buildup either in the surface of the growing medium or in the medium, such that you have to then flush the growing medium to just reduce the EC in that medium. So foliar allows you to, we think, this is not 100% science yet, but reduce your soluble salt inputs by using these supplements, and not such that, say your EC target is, 200 parts per million, whatever. We can reduce it significantly without, say, reducing the inputs by half and then applying half by foliar. You can actually do a lot of things. It's not a one-for-one trade-off if you apply by foliar, just that the plant can access these nutrients more efficiently, and it helps, again, uptake of nutrients from the soil too. So there's a lot of benefits all around. And if whoever put the question in is interested, please, go to the BioWorks website and do some reading on guard calcium, and then follow up with a call. Be happy to talk you through the process.
Okay. You might be able to share your screen if you wanted to go back to any of the slides.
Yep. Only if you let me.
I have let you.
Ooh. Do you want me to? Well,
it's up to you.
We are.
If it'll help facilitate the answer to the question, please feel free.
All right.
Okay. So next we have a question about plants coming from another facility. This producer is saying that they constantly get clone trays full of bugs. What is the best treatment? This is
Legal
a very big question.
Use another propagator. At some point, I think there's got to be a conversation between the people receiving the cuttings and the people sending them. So say, this is just not acceptable. I realize in the real world, that's not always possible. You have to deal with what you get. But I think more attention should be paid to propagators. You know, in the ornamentals world, we've had this back and forth with the major propagators. I think they're doing an awful lot better job now than they ever had in the floriculture side. It will eventually, I'm sure, transfer to cannabis as well, cannabis or hemp even. What can you do if you constantly get them? If complaining back to the propagator should be part of your response, I think. I would always advocate dipping those cuttings before sticking them. Unfortunately, Sufoil X, it's approved for dipping floriculture cuttings, but it's not yet approved for dipping cannabis cuttings. We know in floriculture that it does an absolutely fantastic job against aphids, mites, and thrips, and even eggs of mites, even eggs of western flower thrips that are laid inside plant tissue. So, but it's not approved yet by Health Canada as a dip. So what does that leave you with? There's two options. Botanigard is one of them. I'm not sure about the Biopesticides label, whether it's approved yet for dipping. I think it is, but the Bavaria Bassiana products, let's just say, but check the label. I would definitely dip in Botanigard. I would highly recommend incorporating insecticidal soap in with that as a wetting agent, because insecticidal soap, again, that might be going off-label use. You have to check the label for insecticidal soap. I'm sorry to get sort of into the label stuff, but it's the law. If you can use insecticidal soap, Botanigard plus about 0.5, certainly no more, volume concentration of insecticidal soap will result in a significant reduction of aphids and thrips, but it doesn't really do a whole lot of spider mites. And then root aphids, whole different ball of wax there, and that's where the early drenching of the once the cuttings are stuck, if you're using rooting cubes, and a lot of growers do use rooting cubes, you know, allow those roots to develop about 7 days, then go in with a Botanigard or a Bioceres drench. Thoroughly wet that cube. Again, if you look through the presentation again, I do touch on this in the presentation, but apply it a week, 5 days later, apply it again. The reason for the repeated application requirement is that you're not going to get 100% contact and kill from one application. You're always going to be some survivors, so you've got to do it again, and reducing that interval between applications means that you're not allowing aphids are ridiculous in the speed at which they can increase populations. All of the big pests are, you know. So by shortening that interval between applications, you make sure you're really knocking down the population, not allowing it to build, knock a few more, build, knock a few more down. So you really want to reduce the interval and potentially use a high rate as well for a finite time, and then go back to a lower drench rate, longer application interval. So that's root aphids.
Okay.
Should say and add the if plant pathogens are a concern, that's where the hydrogen peroxide dip, use of xeritol as a dip, and that is on the label, comes in. That'll basically eliminate any microbes on the surface of that cutting, that clone. Doesn't work systemically, and that's where I would highly recommend, again, send some of that plant material off for testing, because if that disease is already inside that plant, there's nothing we have available to us that is going to cure that plant. Even in conventional production systems, and not talking cannabis, there's relatively few products will cure a plant. There are some systemics which work, and that's because they're systemics. We don't have anything like that approved for use in cannabis.
Got it.
Mm-hmm.
Okay, so we'll just keep working our way down. Um,
This is going to take till midnight, isn't it? I'll be less verbose.
that'll be part 3, folks. How do you feel?
We start
How,
charging for the next one.
how do you feel about using UVC as a disinfectant for sterilizing rooms,
Yeah,
plants, or on
yeah.
people?
I think people know. Yeah, UVC can work. I'm not super familiar with it. I know they have been testing it for quite a while in Ontario. I don't know how well it works. It seems that it's one of those things. I think we all want a silver bullet. Alas, there isn't such a thing as far as plant protection is concerned. So it might be part of an integrated program. I know they're testing it currently in Cornell and University of Vermont as a means of, you move the UV lights over the crop. Typically, they're like on a gantry system. And certainly they will help reduce levels of powdery mildew spores that are present, other fungal spores which are just floating around. So yeah, they can do an okay job. But again, it's not let's do this and then we don't have to worry about anything else. But it is potentially something in a specified area, like in a propagation room or in the mother plants. It's something to think about because it's a relatively small area, so the costs are not so high, and you're at that page. I say, mother plants that's your investment. That's the bank you keep going back to. So you really want to look after them.
It's a different kind of plant parenting right there. Okay. So, has there been literature that suggests reductions in cannabinoid production due to promoting salicylic acid defense pathways?
That's a really good question. I can't give you an example off the top of my head, so I'm not even going to try. We do know that a lot of plant stress responses can result in increased production of certain desirable compounds in a plant, but I don't know specifically. So I'm sorry, I don't have an answer to that one. I'll put it on my to-do list and find out.
All right. Next up, cannabis aphids. Those producers saying that it's their biggest problem. Using Cephalosporin X at 2% has not been effective. Do you have any suggestions? And they've also been using both eggs and larvae of lacewings.
Mm-hmm. Yep. 2 suggestions, potentially. Soft at 2% should kill cannabis aphid. The problem, as I mentioned earlier, with all aphids is they reproduce parthenogenetically. The mothers just keep pumping out live young so quickly. So to get that under control, you really need to hit them hard, and that's where I think soft oil can do a job. Insecticidal soap is something else which can work, and maybe soft oil soap in rotation. Spray coverage is probably one of the most underrated aspects of applying a pesticide, though, because aphids typically are found on the underside of plant leaves. So you have to get sprays to where the aphids are. And they're typically, if this is a leaf, here they are, right under here. Where are you spraying from? Down like this, typically. You're getting great coverage on top of the leaf, but that product is not going to move through the leaf and get the aphids underneath. And that's why having good sprayers, if you can get nozzles which target that spray upwards and into the canopy, or at least sideways into the canopy, that can really, really help. And that's where I think some of the foggers can come in, but then you can't apply soft oil, and I don't think you should apply soap either through a fogger. So nix that as an option. So getting a hydraulic type sprayer, which can put out fine spray droplets rather than a heavy wet spray, is probably going to be better. Because those fine droplets will move into that canopy better. You get better underleaf coverage. You get better underleaf coverage. You should get better control of the aphids. So again, it goes back to investing in good spray equipment, I think is probably so important. The other thing, lacewings can do a lot of feeding, but also getting them to stick around in a crop sometimes is really, really hard. So that they will, you can introduce them, they wander off, they find an infested leaf, they'll feed away for a little while, then they'll move on elsewhere. Even if they leave 2 or 3 behind, that's the nucleus for a whole new population growth there. So that's not always the best way. I think perhaps a combination of lacewings with aphid parasitoids can be the way to go. And again, I think for cannabis aphid, I think it's Aphidius urvi is the better one to use for cannabis aphid.
Yeah, you did mention that in your presentation.
Yeah, and it's super important to choose the right Aphidius species for, here we go, for the aphid in question. So anyway, yeah, Aphidius urvi is the one to use for aphids, but yes, here we go. Cannabis aphid, potato aphid, foxglove aphid, Aphidius urvi. And then again, it will establish, should establish in the crop, but you have to release them every week, and there's a rate specified in any guide you get. And it's the release rate is in the presentation. Do you want me to share my screen quickly for that one?
Sure, yeah, let's do that.
Yeah, because we can talk to some of the other stuff. Okay, so cannabis aphid, these are what they call large aphids. So Aphidius urvi, excellent aphid parasitoid, works against all of these. And that's where identification of your aphid is so important, because you want to be sure, and cannabis aphid is quite distinctive in its morphology. The other predatory species is Aphidoletes, and that's in the bottom of those 2 pictures there. Those little pinkish colored and the large sort of yellowy green colored maggots are Aphidoletes immatures, and they very neat. They attack aphids and they sort of puncture their legs and basically use the leg as a straw to suck out all the body contents through that, and the aphid will slowly go down and dry down. That might be something that works as an alternative to lacewing, to Chrysoperla. If you're using biocontrol agents together with the soap and oil, good coverage. Between those, you should get control of aphids, but it is problematic. And I fully appreciate the challenges in cannabis crops when they're so dense and the foliage is so dense and the plants are grown so closely together. That is a real challenge. This is probably one of the biggest challenges out now are aphids to control in cannabis. So
these are some amazing action shots, by the way.
They're good, aren't they? Yeah.
Okay.
Yeah, but again, it's a work in progress. I mean, a work in progress, that's the wrong word to use. It's a process. You have to release these parasitoids early, and they will become established, should become established over time. So it's a constant reintroduction into that greenhouse. But again, it's understanding not only your pest, but understanding your biocontrol agent as well. Is right. What conditions does it work well under? Temperature, light, humidity. What conditions does it not work well under? Are you providing the right conditions for it to work well? So these are all things rather than just buying something because somebody says so or throwing it into a crop. You have to get the right environment, the right natural enemy, and have identified the pest correctly. So those three things have to be done right for biocontrol to work really, really well.
If you find signs of disease, how much time does a producer have to react to it before it becomes damaging?
I would respond this way. If you can see signs of the disease, it's probably already too late. Because symptoms, typically the infection is there. Once the plant really starts to show those symptoms, it's often too late. And that's why you may not see anything, but it's important, just make a leap of faith, if you will, because we've shown it so many times in other crops. The disease will be there. It will arrive. Assume that. Take preventative steps, because all of these preventatives work best when populations, be it pest or disease, when those populations or the inoculum levels are low, and they will provide protection to the plant. And a lot of the materials that are available, I'll go back for the root diseases, for example, RootShield and Pre-Stop, for example, they can be applied at very cost-effective rates. And then it's not just you apply it and it's gone in 2 days. These are living organisms which you're applying, and they will colonize the roots, and they'll give you anywhere sort of from, let's say, 3 to 8 weeks of protection. So it's not like you're applying every week. You can apply through a cannabis production cycle. You may apply twice, 3 times. If you're taking care at propagation, I would go there, stepping up, and then once again. So maybe 3 times in that whole life of that crop. It's again, what's your crop worth? If you let that disease develop, 2 things are going to happen. One, the plant's going to die, or the best case, production and quality is going to be compromised significantly. The longer you leave that diseased plant in a crop, if it comes to it, you're just risking infection to everything else that's around it. So I would say prevention is one thing. If you start to see disease, what actions can you take? Sometimes it's too late. The only disease where you can take curative action is mildew, but again, be curative early in that disease development, and that's where using the potassium bicarbonate sprays, Milstop or Sirocco, can work well, because they have preventative rates and curative rates, and it's, you can go in and cure a crop, but that's probably the only curative tool that we have for cannabis right now. If nothing works, if you're too late, then the only recourse, I think, is actually roguing plants out as well. And that's hard to do. Oh my gosh, it's hard enough with guys throwing out, bedding plants, for goodness sake. I can only imagine what it's like if you have to, take out, and take it out as cautiously as possible. But sometimes roguing is the only way forward. And that's, there are quite a few viruses starting to show up in cannabis now. No cures for viruses. The only thing you can do is remove it. So you're removing source of infection for the rest of the plants. But, again, taking care of that right up front, preventing diseases happening right up front. And it goes again, how many times did I say it? Prevention, sanitation, and it applies across the board. And those are the best tools that we have. Those are really, in many respects, the only tools that we have for cannabis production.
Okay.
Sorry, there's no better news there.
No, I mean, the best method is prevention for many things, even outside of greenhouse production.
Ounce of prevention is worth a pound of cure. You've heard that how many times?
Have you seen evidence of Fusarium graminearum in cannabis inflorescences?
Personally, I have not. I'm not saying that it doesn't happen. I've not seen it.
Okay.
Fusarium is one of those funny organisms. You can see it, as a soil-borne disease, as a disease of in the stems and things like that, and then some maybe in the flowers. All I know is a lot of fusarium species have very wide host ranges. Cannabis to them is, oh look, here's another host that wasn't previously grown necessarily, but now it is. If it's out there, it's going to come in. So again, what's that P word? Prevention. Yeah. So it could be. I don't know. That's something I would have to do a little bit of a deeper dig into to find out that information.
What would you suggest for botrytis control in flowers?
Botrytis is a hard one. There's nothing registered for control. There are several options registered for suppression. And I can, whoops. If you want, I'll pull up that slide again as well. Okay, flowering to harvest. This is the tricky time, because with the pistils and the trichomes in the flowers, that's where the good stuff is. So you don't want to risk burning those. And we know that certainly at high rates, some of the potassium bicarbonate can damage trichomes. It doesn't always. It's probably linked to temperature and all sorts of environmental conditions as well. We have had growers use potassium bicarbonate, Millstop, and Sulfoylex practically up until harvest with no adverse side effects. But I would say you've got to be real careful about using those. So our general recommendation is to not use them within 3 weeks of harvest. So that still leaves you 3 weeks to either bite your fingernails all the way down, or think about, well, what else could I use? Is there really the only materials that can provide some degree of botrytis suppression? There's the Regalia max, and that's the one that activates the plant's own defenses. Good product. Does it translate into the flower? Not sure. RootShield HC, again, this is trichoderma, foliar spray, but again, and ditto for pre-stop, that's the Gliocladium. Stop those sprays 3 to 4 weeks, 3 weeks prior to harvest, because you don't want to just be putting microbes into the flowers, because they will show up in the test. Millstop Sirocco, that's potassium bicarbonate. And this is where, back to the first question about the plant supplements, this is where On Guard calcium, we think, could help in this regard as well, because it gets down to enhanced calcium uptake by parts of the plant which typically have problems moving calcium into the cell walls, so the cell walls can be weaker. Botrytis loves to go into compromised tissue, to senescing plant tissue. That's where botrytis gets a foothold. So the healthier you can make that tissue, the more tolerant it's going to be, the more able it is going to be to resist infection by something like botrytis. And then the other tools can help on top of that. So I think nutrition in cases like this is probably part of that prevention program again, rather than sort of pushing plants too hard to get bigger flowers. And then we know that's standard practice in many cases, understandably again, but it oftentimes leads to weaker cells. So if you can cut back on some of the nutrients, is that realistic? Maybe it's not. So it makes it even more important then to get some of those nutrient supplements to improve calcium uptake by those plant tissues. And that's why I don't know if everybody saw the announcement by Health Canada around being able to apply foliar nutrients now and apply certain products by foliar spray. It's an important ruling that has a lot of implications to cases like this specifically, where where you need to get extra calcium into those susceptible tissues. So there's no one way, I don't think, that is going to get rid of botrytis. Again, all of these can help prevent, suppress botrytis happening, and that's what we can do. Nothing is going to cure it.
All right, so we're going to wander back into BCAs.
Wonder.
In terms of an extreme infestation of spider mites, do you have a recommendation in terms of how dense the predatory mites should be in terms of per square meter?
There are cases where I would say that's where using a spray, something like Sofoil or Pure Spray. I'm being partisan because I think Sofoil is a really good product and it does work super well against mites. Why would you use both? If the populations are so high, it's, again, getting back to, you've got to use multiple tools together in an IPM program. But the predator on its own will be doing a great job perhaps, and it's eating, tens of spider mites every single day. But the population is so huge that it doesn't really bring it under control in a timely manner. And that's where you need to use something like a Sofoil spray to really knock those pest populations down. And maybe even do 2 or 3 Sofoil sprays, knock the populations back, then introduce your Persimilis into those hot spots. Persimilis is a phenomenal predator. It really, really does a great job, and it will clean up hot spots. Obviously, the sooner you get into those areas, the better results you're going to see, the less treatments you have to apply. So again, it's a matter of getting into the crop, scouting. If you're starting to see webbing, that should be like, oh my God, I've got to do this now. There is no time to waste. If you've got thick webbing, like spooky movie, Halloween spider webbing all over your plant, good luck. Again, I hate to say it, but it might be time. It's too late. Yeah. What could you do then? Just remove the plant, but still follow through with those Sofoil sprays with the release of Persimilis. But honestly, the key is to try and prevent that happening, and that means good crop scouting, regular crop scouting, an ability to recognize what spider mite feeding damage looks like on a leaf, and it's quite distinctive. And then don't wait a few days. Take steps immediately if you need to. But again, the whole point of the preventative program is to stop that happening. And don't get it's easy, I think, sometimes to get comfortable. Oh, it's working. You walk away. But spider mite's another one of those pests which can almost quadruple overnight, it seems. And that's a ridiculous rate of increase, or the potential rate of population increase. So for every day you wait, it's already doubled its population. So don't. Take steps. I think one of the points I was trying to make at the beginning as well is planning. You should have that program planned before anything even goes into a pot or a cube or anything like that. Now is when next year's production plans should be being made, so you know what to do. Ask those what if questions. What are you going to do? And it allows you to be more proactive, and I think in sort of knowing what you're going to do, more proactive in terms of understanding what you need to do, where you need to go, and the tools you need potentially to bring in to take care of that situation. But the thing with any biocontrol strategy using predators, you can place an order today. It doesn't arrive tomorrow. It'll arrive, maybe 4 days, 5 days, a week later. So that's another factor you have to build in. Within time of order, time of receipt, you know, say you've got 5 days goes by in a life of a spider mite, that's like, oh, yeah, we've had kids, the kids have had kids, the kids have had kids, the kids have had kids. Boom. So you've gone from a small problem to a large problem very, very quickly. So preemptive planning is always a good thing to do.
Okay. We're going to head into our last couple of questions. Do you think the success rate of Aureus improves with the use of supplemental nutrition, such as Aphestia eggs?
Yeah, I do. Nutrimite's a good one as well. That's a pollen supplement. There's Aphestia eggs, and there is, I'm not sure if, I don't think you can get these in Canada, so I'm not, no, I don't think you can. Aphestia eggs and Nutrimite, I know you can, which is pollen from Tifa, which is cattail, and Aphestia eggs work beautifully. The nice thing, Aureus is a generalist, so it'll, if it can't find a prey organism to eat, it will feed whatever it finds. If that's pollen, if that's Aphestia eggs, yes, they will establish better. Essentially, you're providing resources to our Aureus to stick around, to encourage Aureus to stick around, to encourage Aureus populations potentially to grow as well. But Aureus as well, you know, the time of year you release Aureus is going to have a huge bearing on whether they will establish or not. They do typically go into some sort of diapause once the day length gets below a certain number of daylight hours in that day. And I mentioned this, like 4 to 5 months of the year where Aureus just doesn't want to know. So it's one of those things as well. Yes, the supplemental feeding, also use of banker plants have proven to be very, very successful too. But banker plants is not my, let's use banker plants first. You know, that requires a degree of experience, I think, in use of biocontrol before you can start to use banker plants. Once you start to use them, they can be hugely effective in terms of providing a consistent supply of natural enemies like Aureus into the crop, providing a place where Aureus can breed and increase in the absence of prey potentially in the crop. So, but that's, you know, once you've gone through Biocontrol 101 and then the advanced level, it's the next level to get into. Certainly, we would love people to get to that point, and it can work superbly well in many crops, and cannabis no exception. But it does require a whole different level of organization and planning, because you don't have to now just grow your cannabis crops, you have to grow your banker plants as well, that have to be flowering at the stage when you want to introduce them, and you have to be able to have a constant supply. So now you're producing cannabis, you're also producing banker plants as well. So there's a whole timing process that you have to think through as well. So it is a little more complex, but it can work very, very well. So, I mean, that's the ultimate. In the interim, what the supplemental foods do is they're just, in place of a banker plant, you can put supplemental foods into the crop. Yeah, so they can help.
Now, do the
God,
supplemental...
that was a long answer for an easy yes/no question, wasn't
No,
it?
I think you brought up a great topic, banker plants.
Mm-hmm.
Do the supplemental foods have to be approved for specific crops?
Good question. Don't honestly know. Part of me says I don't think so. Why would you? But maybe check it out. Yeah, I would place a few calls to my plant product supplier and ask them, to the biocontrol expert, and say, can I put pollen in? That would be a call I would place to them, or Terralink in that manner.
Got it. Okay, last one, just to wrap it all up.
Yep.
You know, the webinar was very focused on preventative steps, and there are so many and so many different products.
I was going to say, be polite, Greta.
Well, there are just so many options and so many preventative steps, but how would a producer know which ones to use in advance of the problem, because they don't know what the problem's going to be?
Yep. I think and this is part of the problem, you know, because cannabis production at scale, at the scales we're growing at now, is only what, 2 years, 3 years old now. So we really don't know. We don't know. This sounds Rumsfeldian, but we don't know what we don't know yet. We're still in that, I think, learning process, very much so. But I think there's a lot of lessons can be just transferred from what we've learned in floriculture, just other flowers, let's face it, can be transferred over to cannabis, you know. And I think the understanding is that, you know, you will get pests, you will get diseases. If you accept that, and prior experience, even in cannabis, short as it is, I'm sure many growers say, oh yeah, we have this, this, and this as our primary problems. Are you keeping good records? Do you know when this problem showed up? Do you know where that problem was? Did you get it in your propagation room? That says to me then, you know, hey, we need to tighten up procedures in the propagation room. Was it at a certain time point through that production period? Was it at a certain time of year? All of these pieces, if you look at it as sort of like a mystery, you start to piece these together and you get a clearer picture of what pests you have and when, what diseases you have and when. And with that knowledge, you can start to plan your IPM program around it. Or you make assumptions that certain pests will occur. If they don't appear, then you can plan for it, but you don't have to sort of pull the trigger and do it. I would say 3 things to count on. You're going to get aphids. You're probably going to get some form of mite. You are going to get powdery mildew. You are going to get root diseases, be challenged with root diseases. Make those 4 assumptions, plan around that with contingencies around some of the other pests, perhaps. And then go full steam on that preventative program. If you don't have pest or disease issues under that program, you can do one of 2 things. One is say, why did I do all that? I didn't get pests and diseases. But maybe you didn't get pests and diseases because you were doing that. So don't stop doing everything until you know. But once that confidence comes of using certain things, I think that's the point at which you understand to some degree crop dynamics, how they associate with pest dynamics, how they associate with climate, with environmental controls, etc. You may start to be able to pull out some of those pieces. It's like a Jenga puzzle. Which can I pull out and it still stays standing? But do it piece by piece. This is how the you know, Ontario's floriculture industry, for example, has they're you know, light years ahead in my view of many countries in terms of the sophistication and the degree to which they use biocontrol, because of the experience, and that let's use everything, and then we'll stop you. We'll little by little we'll see what we can do without, and that's the only way forward. You know, it's there's no quick and easy. You know, I wanted to, without going into an absolutely exhaustive list of all of the biocontrol options that are potentially out there, I wanted to give representation to the ones which we know work, the ones which we know work in cannabis, and the ones which we know sort of play well together and aren't too badly affected by the biopesticides that we have at our disposal. Or if they are affected, we know how we can go back in and reintroduce them. So, and I think that's what biocontrol is all about. You don't have to use all of them. Those are the options. And I think it's every location is going to see success with some things, failure with other things that work really well in another location. It's a matter of finding what works for your specific location, your specific cannabis varieties, your, you know, whatever else, and make it work for you. And that's where there's anybody that says, you know, here's how you can do it, I think is to some degree not telling you the whole truth. There are certain, I would say, foundational elements I would put into a biocontrol program, but it always needs consistent tweaking to make it work as well as it can work in a specific location.
Well, thank you for that.
Pleasure.
There are still actually a number of questions, but I'm actually going to send these to you because they are more product focused about specific use of the products or compatibility. And then that way yourself or a team member can follow up with them
Certainly will do.
personally.
Thank you for that. Yeah.
Okay. Well, thank you for joining us.
Oh, you're
And
welcome. So here's a pun. Will this go on as an annex to the main presentation? Did you get that one? That's a good one, eh? Shall we explain that one while we're still on air?
Because Greenhouse Canada is owned by Annex Business Media.
Ta-da! Anyway.
If only I had a logo.
I know I should have one behind me, but need the green screen.
Yes, yes. Okay, well,
Alrighty.
have a good one.
Thank you, Greta. Pleasure, as always. And one of these days, I'll see you outside of the confines of a Zoom meeting or a go-to.
Here's hoping.
Here's hoping.